So I'm going to talk about the work we've been doing improving the accessibility of over 100 billion genetic associations. So brief background, this is predominantly based on genome-wide association studies, which is an approach used in genetics research to associate specific genetic variations with particular diseases or traits. So you take two populations, perhaps cases and controls, and identify the regions of the genome that are associated with a particular disease, perhaps. Over the last few years, we've been collecting these data into batches which have a shared origin, so perhaps a UK biobank. And then due to constraints on the number of records in an index, we've grouped these into 500 per batch. Each GWAS of those 500 contains around 10 million genetic associations, and each individual record looks as shown on the right there. So we have around 10 fields, a mixture of keywords and integers and floats. And so the actual data itself is relatively simple. It's just there's lots of it. So we were fortunate to start working with Oracle a few years ago, which led us to experiment with various configurations of a cluster. And we're currently using 10 kind of basic VMs with 32 gigs of RAM, 16 CPUs, each with over 6 terabytes of block storage. And the main query types that we're using across this data, the vast majority are just looking for a small number of records identified by a GWAS ID and a SNP ID, so that's just 2 IDs, and perhaps a range query on a p-value. We also have the option to do chromosome and position range queries. And there's quite a lot of queries that look over the entire data set by a single SNP ID and then a p-value. And these vary in how many records they return, the performance and the requirements of each of those. So the first lesson we learned was quite an obvious one, that you should use keywords for term queries and not integers. Naively, I assumed because the GWAS ID was an integer, we should use an integer type, but term querying with integers is slow. Keywords is fast. So that was the first thing we learned. The second thing, we had a live Elasticsearch cluster, a local one that we were using initially, but we were also trying to index simultaneously whilst providing it as a service. And this didn't work. So we moved to bulk indexing offline using HPC. So that actually ran in over 1,400 cores in 24 hours, and we could index everything, create snapshots, and then copy them onto the cluster and restore them without any impact, really. The third thing, which was pointed out at a Elasticsearch meetup, very kindly, was to use post-filter. So instead of running one filter query over everything, was to use a filter based on the term query first, and then run the more computationally demanding range query on that afterwards using post filter. And this saved us a lot of time, which was great. Next lesson we learned was to use multi-search. So we only just started using this recently. Our queries were actually looking over each index batch sequentially, but that was actually very, very slow at times. And then I started using multi-search, which meant this was actually running in parallel, which had a massive increase in performance. That was, and I guess the big question is, what has this provided us, and how has this improved the accessibility of this data set? We're now providing this through a platform called OpenGWAS here at University of Bristol. We have over 34,000 data sets, currently around 125, 130 billion associations, and an increasing number of API calls per day, sometimes up to 5 million or so. And the changes that I've discussed and the improvements that we made provided a massive increase in performance. We can now do sub-second queries for quite complex, large-scale queries across the entire data set. And we're now the world's largest and most performant collection of genetic association data, with an increasing number of users and API calls. Lastly, just acknowledgements and links.